The origin of life is a complicated but extremely important process. It's easy to misunderstand how it happened, and I misunderstood so many things about it initially. So here are 7 things that most people get wrong that you will now get right about the origin of life. Let's dive in. Hey guys, welcome to the channel. My name is Reese. I used to teach tech ethics at MIT, but now I do this series on how everything evolved, where I kind of synthesize a decade of learning in these 20,000 note cards I've done, and I want to share those learnings with you. Today, we are talking about the origin of life. And the crazy thing about the origin of life that we all know, if you imagine this little sheet here, we're talking about, well, my cat stepped on this recently, so he made it bad. But we're talking about this very beginning period here. How did life begin? Very interesting, very, very interesting. That small piece there is actually very complicated, obviously. So the first thing that people get wrong is they say, we don't know what life looked like back then. Actually, we do. It's called the Last Universal Common Ancestor, or LUCA. In the last decade, we've learned a bunch about LUCA. So here's this amazing page about LUCA, the Last Universal Common Ancestor. And as you can see, if you go back in time, you say, oh, what was it like before humans and before even with bacteria and archaea? And you get to this last universal common ancestor, the little creature that everything came from. And so that LUCA thing, there was a study in 2016 that looked at, 300,000 different protein clusters from all these different prokaryotic genomes. And they looked at these 6 million different genes, and they found these 355 protein clusters or genes that were probably common to LUCA. So yeah, we know a lot about LUCA actually. It looks like this roughly. It was probably a hydrothermal vent. It was probably an extremophile, or a thermophile rather, that it needed to be in over 80 degrees Celsius. And it did a lot of the things that we would expect around proteins and cell division and ribosomes and carbon fixation, nitrogen fixation. It also used, interestingly, the WL pathway, the Wood-Lungdahl pathway, which was one that it produces 1 and 2 carbon chains, a lot of acetate. And so here's that article, the 2016 article, The Physiology and Habitat of the Last Universal Common Ancestor. Great article. And it shows, hey, you can look across all these bacteria, across all these different kinds of things that they might do, like ribosomes and whatever, and DNA, and which ones do they all share? And then you can start to construct this little cell back then. The other article is a more recent one from July 24, so about a year ago. And this says, the nature of the last universal common ancestor and its impact on the early Earth system. And this one is kind of an update to that one. You can see they're constructing a LUCA back in time. And here's all, they're doing this maximum likelihood thing where they're like, oh my God, let's look at all these different creatures. And let's look at all these creatures and let's see what the heck do we think this thing was. And you can determine from it, I love this image, but you can see here, this is metabolism. This is the universal metabolic tree or the universal metabolic network. And the black lines are showing what metabolism LUCA had. So that's amazing. And the other amazing thing is you can see here's their version of LUCA. It's pretty similar. They also say some really interesting things that it was LUCA, but there were also a bunch of contemporaries. There were a bunch of other creatures around at that time. And interestingly, they even show that LUCA probably had a Cas-based immune system, so it had a little bit of immunity to other cells or viruses around it. So people say we don't know what the first life was, but we do. With molecular genetics and molecular phylogenetic trees, we can actually determine what the first life was, and it's this LUCA. So beautiful, so amazing. The second thing that people get wrong is they think there was just one LUCA, Last Universal Common Ancestor. But in fact, that's wrong. There were many LUCAs. There are all these little creatures here, and they were interacting in an ecosystem with each other. So you can see this here, B on the graph, where it says LUCA and contemporaries. Because if you're a little LUCA, you're outputting little chemicals, and the other ones are eating it, and vice versa. And you're all similar to each other, and all these little protocells are budding off of each other, and you're interacting in a network and a community and an ecosystem at the beginning. And we can see this later in time because this is a beautiful thing called a microbial mat. It is, when we see microbes today in the world, often we see them as this little series of creatures that are kind of stacked on top of each other. And this is, millions and billions of little creatures that all exist in an ecosystem together. It's like a little city or a little coral reef, but a little microbial coral reef where the ones at the top are kind of doing the oxygenic things and they can work with oxygen. And then down below, those are the little creatures that they don't like oxygen, but they can consume things from the top. And you can see here's an example of a microbial mat with all these different kinds of little creatures in there. Or here's another image of a microbial mat with all the different compounds and minerals and sulfur and carbon dioxide that they're sharing with each other. So if you imagine all of these little creatures as little LUCAs, there wasn't just one LUCA, but there were many LUCAs. Isn't that cute? Look at all these little fluff balls hanging out together. Okay, the next thing that people get wrong is they think that LUCA was the last, the very first thing, but in fact that's wrong. It was actually the final stage of something earlier than it called the progenote. And so to understand the progenote, you can imagine here's all these little LUCAs interacting with each other, and back then, before they got more cellularization, before they started to be differentiated from each other, they were existing in kind of a big soup, a big mess. And we call that mess the progenote. Here's a picture of the progenote. You can see, here's LUCA. We have what we now know of today as vertical gene transfer, where I give genes to my kids and I got the genes from my parents. And so we can kind of go back in time 4 billion years all the way to LUCA and say, there was the first creature, the last universal common ancestor. But in fact, before that was this thing called the progenote, which was a set of these little protocells with proto-RNA, proto-DNA, all kind of working with each other in collaboration. And at that time, there was what's called horizontal gene transfer, where they're all just kind of exchanging genetic information, RNA or DNA, with each other. This was before they individualized and before we got vertical gene transfer. Back then we had more horizontal gene transfer. Many prokaryotes still do today. They still exchange genes in a horizontal way, but they did it even a lot more back then. And Carl Woese, the guy who came up with this, calls this the Darwinian threshold, where only after this point do we start to get what we traditionally think of as DNA-based heredity. So I like to think of this using the frame of common pool resources, which is a thing that people talk about today when they say, if you have a fish pond, that's a big common pool resource of fish. And if you just have people all access that common pool, then you get stuff like overfishing, which we obviously have today. And so early cells also had this issue. The common pool resources for them were things like ribosomes, the 3D printers of the cell. It was things like RNA, where they had little bits of code information. They had metabolism, where that was like kind of the energy or biomass. And so if you're just sharing all of your energy, all of your biomass, all of your 3D printers, then people can kind of be cheaters. People can come in and they can overfish that. They can say, I'm just going to use all these things. If you're sharing everything with all your friends, that's an issue. And so what they did is they made this all a club good. And a club good is where you take a resource and you say, no, only some people get to use it. And so you have these little creatures that say, you know what, I'm going to start to be individualized here. I am going to not allow other people to steal my metabolites or to steal my, these new ways of these cool new RNAs that I have. I'm also going to start to spend more energy on my RNA. I'm going to turn it into DNA, a thing that costs more energy to maintain, but is more stable because it's this double helix instead of the single helix. This ribosome that I use to print, RNA is the input and proteins are the output. I'm going to make a standard there. I'm going to standardize, just like we all standardize around TCP/IP or, the 26 letters of the alphabet. They're going to standardize around this coding scheme. And so other people can't, so there's this new standard coding scheme that's used for everybody. And so once you had the progenote where everything was together, and then you start to get a little bit of LUCAization, where these things start to be individualized. They start to say, no, I'm going to not just have a protocell, but a real cell, and I'm going to be different than you. This was essentially equal to the enclosure movement, but for cells, where, this happened in Britain in the 1700s, where they said, hey, everybody can't just be on everybody's land. We're going to make private property here. And this was actually very helpful for getting more returns out of crops. It had negative ramifications too, but back in the day, progenote times, this was everybody saying, hey, we're going to enclose our own things. Everybody doesn't just get to use everybody's resources. The next problem that people usually have is they think that the ribosome in these little creatures, these little LUCA style creatures, was just a thing, just a part of the metabolic machinery. But in fact, the ribosome was everything. I'm definitely a ribosome maximalist. I'm a Krebs maximalist, and I'm a ribosome maximalist. So as a reminder, what is the ribosome? The ribosome is this little 3D printer in the cell. And what it does is it takes as input, that little bottom strand is RNA, and then it's taking that RNA and it's turning it into proteins, and those proteins eventually do work. And so the ribosome is a little 3D printer that takes code as input, RNA, and outputs objects, these proteins that actually do work in the cell. And here goes, oh, it's going to take that, a little bit of protein, and it's going to move it somewhere. And so this is another picture of the ribosome. It's this little 3D printer in our cells. A given cell might have 1,000 mitochondria, but it'll have like 1,000,000 ribosomes in it. So they're printing all these little molecular machinery. And to understand the ribosome, here's a good picture of it. What it's composed of, it's about 2/3 RNA and 1/3 proteins. And so I believe that the orange, the kind of orange, I don't even know what color that is, like pink? The orange, pink, tan color stuff is RNA, and the purple are these proteins. The ribosome, so it's composed of a bunch of folded matter, RNA and proteins. And so I like to think of the ribosome, this molecular machine composed of RNA and proteins. And I like to think of it as kind of the final form, what's called the folded matter phase, where you had little bits of RNA that were saying, whoa, what is it like to fold? How can I fold? What are all these different ways I can fold? I can fold like this. I can fold like this. If I'm a little protein, oh, here's how I can fold. I can fold like this. And then eventually these things, they're stable forms. You have to stick around first. So you have to be stable, and then you can start to be useful for the system. And so these things are trying to be stable and useful. And eventually what they form, there are two things that they form. You can see that the ribosome is actually composed of two units here. One of them is called the large subunit, and one of them is called the small subunit. I'll let you guess which one is which. And the large subunit, its main role is that it does peptide bond formation. So it creates longer and longer protein chains. It brings amino acids together to create protein chains. And then the small subunit, what it does is it does the translation piece. So it's kind of taking as input an RNA code and outputting which proteins to hit to each other. The crucial bit is that each of these evolved independently. It was helpful to make long chains, to be a large subunit, to do condensation catalysts. I'm just going to make more and more long protein chains, and maybe those will help the system. And then it was also helpful to be a small subunit. It was like, oh, I'm going to be a translator between these two worlds. That was really helpful. And then you combine them together, and they say, oh, if with our forces combined, we can do amazing stuff together. And so this is a key theme that you see throughout the origin of life, throughout humanity, is things need to work by themselves first, and then they can work in combination. My iPhone is not just the screen. It's the screen that works in combination with the GPS, that works in combination with the camera. And so they all work well, and then they work really well together. But you have to have the stable subunits first that can then combine with the other modular subunits to create a bigger process. Here's this other amazing image of the ribosome. I love when things spin like this. I'm a simple guy. And so the final thing I want to say about the ribosome is that if you imagine each of these as little ribosomes, one of the crucial things that the ribosome did, it started to say, oh, here's some high value proteins that I'm going to start to deterministically create. And of the high value proteins that it deterministically created were the proteins and the RNA that created itself. So the ribosome is a little printer, and the first thing that it prints is more copies of itself. That's why you get 1,000,000 ribosomes in a cell, because the key thing it needs to do is create more copies of itself. This is kind of like how stone tools during early sapiens created more stone tools. Because you get more stone tools, it gets more energy, it gets more people, they create more stone tools. It's also similar to fossil fuels, where fossil fuels were used, the first steam engines were used to pull more coal out of the ground, which allowed you to produce more steam engines, which allowed you to produce more coal. And so at the beginning, all the steam engines did is they produced more coal. And so you have these very tight feedbacks at the beginning of these processes, where the whole goal of it is to rocket ship more of that initial self-reproducing machine. And even with AI these days, one of the AI's main goals to do the lithography, to be helpful for coders, to get more compute, to get more data, so that you can get more, so GPT-5 and GPT-6 can be even stronger. And so the fifth thing that people get wrong is they say, okay, we exist in this RNA world. That's amazing. I think they overemphasize the RNA side of things. I like to think of it more as a more general folded matter phase. And I also like to remind ourselves that there were many, many things before the RNA world. So I think a lot of people think, okay, we were living our life here. How did life start? It started with the RNA world. It's like, it's better to think of that as a more general folded matter phase, where you're just proteins and RNA are trying to see what kinds of conformations they have that are stable, and also what conformations they have that are useful. a survival of the fittest folded matter. You can see that with this image here, which says there's RNA world, but you can also think of an RNP world, an RNA and protein world. And before it, there were just molecules. And then after it, you get DNA and LUCA and everything. I think the other thing that people get wrong about the RNA world is they think it is kind of simple to create RNA. But remember that, and you can see here's RNA. Oh, it looks kind of simple. It's on the right side there. It has a given RNA could have 10 bases or 100 bases or 1,000 bases, 1,000 little letters in it. And each letter is very complicated. Each one has like 50 atoms in it. And so the RNA world is not an answer to how did life begin. It's only a midpoint between smaller molecules and LUCA. The next issue is that people think in terms of sequences, in terms of an RNA sequence. They say, oh, which combination of RNA might be better than the next one or whatever. But in fact, you should be thinking in terms of structure. And so you need to actually think in terms of molecules. I think when people think about RNA initially, they're like, oh, here's this sequence of RNA, and here's C, G, A, and U, in which you can combine them in certain patterns, and those do certain things. And that's true, but the earlier you get, you actually have to get molecular with it. So as a reminder, here's one of those letters. A single letter is composed of 3 subgroups: the nitrogenous base, here's adenine, the sugar, the 5-carbon sugar, the ribose sugar, and then the phosphate group, a phosphorus with a bunch of oxygens around it. And so this is lots of atoms, 50 atoms already. And so instead of thinking in terms of sequence space, that comes much later. That's a different design space that life needed to explore only after it had explored the molecular design space of carbon chains. So before there's a folded matter phase, there's a chained matter phase, and you have to understand who wins in the chained matter phase. And so one of the crazy answers for who wins during this molecular competition phase is this combination of adenine plus sugar is called adenosine, and adenosine is actually everywhere. So here's an image of a bunch of other helpful molecules at the beginning, and you can see the adenosine, the little adenine plus ribose sugar, you can see that everywhere. It's here, it's here, it's here, and it's here. It's all in the red. And so up here, it's like ATP. ATP, the energy currency of the cell, is composed of adenosine. Coenzyme A, another crucial molecule, is composed of adenosine. This thing, NAD+, which is the main electron carrier in the cell, that's also composed of adenosine. So yeah, people think about an RNA world, but they should just as easily be thinking about an adenosine world. And the 7th and final mistake that people make is they say, okay, I'm thinking in terms of molecular structure, that's great. And hopefully they think, oh, how did we get all of these carbon chains to even start to experiment? That's good. That's a great way to think. You say, oh, Krebs, the reverse Krebs cycle, that produces all these 2 through 6 carbon chains, which is true. And that's good, and I'm a Krebs maximalist. But what people don't understand is 2 things. One is that the Wood-Lungdahl pathway was actually more prevalent in early life, and it created the feedstock for Krebs. Krebs mostly works by building up bigger carbon chains, 2 through 6 carbon chains, while the Wood-Lungdahl pathway, the acetyl-CoA pathway, you're creating 2 carbon chains. And so you need a feedstock to feed Krebs initially. That's one thing that people don't think of. And when you look at LUCA, by the way, LUCA has a lot more Wood-Lungdahl than has the reverse Krebs cycle. The other thing that people don't think about is that Krebs is not just the Krebs cycle, but it's also an additional network layer outside of it that helps make the Krebs cycle faster. And these are called the cofactors. So you can see, here's the Krebs cycle. We love the Krebs cycle. And you can see how it builds up. If you're doing this, reverse Krebs cycle, you're taking these small carbon chains, pyruvate, whatever, and you're building it up into 6 carbon chains. That's great. But you also need the acetyl, the 2 carbon chain, as a feedstock for Krebs. And this is the Wood-Lungdahl pathway, which actually produces the fuel that Krebs needs to run, or that rather reverse Krebs needs to run. And the other thing that people don't get, which I find so fascinating, is you can imagine this initial Krebs cycle, and that initial Krebs cycle, you can see in all of these different, there's these little color-coded things, and they say, oh, reductive carboxylation, reduction, H2O, retroaldol cleavage. These things are saying when you're adding on, you need to add on CO2 or take off CO2. You need to remove H2O sometimes. And so at each point of initially this was just kind of happening naturally, but over time, you start to have these little molecules that are helping the Krebs cycle run faster. You need to have something come in and bring in a CO2 molecule or take away a CO2 molecule. You need to have things come in and take away electrons or hydrogen. And so this image shows that. It's kind of a confusing image. These are the B vitamins. The B vitamins are crazy. They're amazing. And what each of them does is, you can see here's B1, B2, B3, B4, B5, B6, B6, B9. They have their names, riboflavin and niacin, you heard that one, or biotin or folate, and they have their little molecular structures here. And the key thing that each of them do is the little blue line here, decarboxylases, or, over here you can see carboxylases. And so I've added things at the top to show what each of them is doing. So, thiamine, it removes CO2 from one of these things. Biotin adds CO2 to something. PLP, B6, it does aminotransferases. It removes or adds a nitrogen. You can see these little FNM and NAD plus or whatever. Those things, riboflavin or niacin, those things add or remove an electron or a proton. So they add or remove a hydrogen. And then, acetyltransferases. This is essentially acetyl-CoA. Here it is. Here's acetyl. This thing adds or removes 2 carbons. And then finally, here's tetrahydrofolate. Folate over here, which does 1 carbon transfer, a methyltransferase, a 1 carbon. And so all of these cofactors, these B vitamins, they were some of the first things that Krebs, once we moved from the Wood-Lungdahl pathway, it was starting to produce, lots of 1 to 2 carbon chains. We eventually started to run reverse Krebs to turn energy into biomass. And so we're doing a bunch of that. We're producing a bunch of biomass. And then Krebs, what are the first things that it starts to create? It starts to create things that help it do its work better. And those things are here. They're these cofactors. They're saying, okay, these are tiny little molecules. They're not as big as RNA. They're not as fancy as that stuff. But what they do is they help double down on the reverse Krebs cycle. They help encourage each of those little reactions to take place. And this is similar to how the ribosome worked, where one of the first things that the ribosome built, it built more, it was able to say, I'm going to create more ribosomes. I'm going to create more things that help me produce more 3D printers. So these are the 7 things that people get wrong about the origin of life. They say, okay, how did life start? I don't know. No one knows how it started. Actually, we do know. There's this thing, LUCA, and we know pretty much all of its genes. We know what it looked like. And they say, okay, there was this one LUCA. It's like, not really. LUCA was part of this ecosystem of LUCAs. There were many little universal common ancestors. And then they say, okay, well, before that, LUCA was the first thing. It's like, no, there's this thing, the progenote, which was doing a bunch of horizontal gene transfer before you eventually got some kind of enclosure movement that had them all start to individualize. Only after we pass that Darwinian threshold do we get LUCA. And one of the crucial parts about that little progenote, and so during this little progenote phase, aka kind of a folded matter phase, where all these different, proteins and RNAs are trying to determine what is the most helpful for the cell. One of the most helpful things was the ribosome. So the ribosome was kind of the final form of the folded matter phase that allowed the progenote to deterministically create high-value proteins and eventually individualize into cells like LUCA. The other thing that people get wrong is the RNA world. They say, oh, how did life start? Well, there was maybe LUCA, and then before LUCA, there was this RNA world. Well, it's better to think of it as a folded matter phase more generally, where RNA and proteins are trying to determine this survival of the fittest folded creature. Then people think in terms of, they're like, okay, I'm at least thinking in terms of sequences. No, you should think in terms of structure and things like adenosine, which were these survival of the fittest set of molecules. And then finally, in order to get those molecules, in order to get the feedstock of carbon chains, you need to create it somehow. And people initially do get the idea of Krebs, that reverse Krebs turns energy into biomass. That's amazing. But they don't understand that there was something before that, the Wood-Lungdahl pathway, which produced the feedstock for Krebs, these even smaller carbon chains, and that also the cofactors were able to double down on the Krebs network in order to make that a more efficient system. So we should think of cofactors and Krebs together. So I hope this helps. We got some nice colors and some nice black and white here. If you have any other questions about the origin of life, please let me know. I'm definitely not an expert. I'm just a casual, but also super non-casual, super serious observer trying to understand how things happen. So I thank you for your curiosity, and I am excited to see you as we actually get into microbial life soon. Thank you, as always, for watching, and thank you to Roots donors for supporting this channel. If you want to understand more about the past and how we got here, check out the previous videos here. And if you want to understand the future and where we're going, feel free to subscribe here. Hope to see you then. Bye.